Thank you so much for the invitation. And I think, these meetings are very important. We will have open forum, and I hope I'll be able to answer your questions. But, I don't see patients. Okay, so if you have very specific medical questions, I may not be able to answer them. I am a scientist, and my goal is to build translational, research so that our discoveries are translated to effective treatment strategies for upper motor neuron disease patients. Okay, so that's my goal. As a scientist, my goal is to generate information and knowledge so that we will understand the disease better and we will develop treatment strategies. Okay, so before I started working, as a professor at Northwestern, there was really not much available at the time. We did not have model systems. We did not understand the genetics. We did not know much about the disease. So having treatment strategies for upper motor neuron disease patients was almost like utopia, like it was unreal, like, it was not expected, right? But within about 10, 15 years, I think we have seen, we have seen the light at the end of the tunnel. And in our research, and also research of our colleagues, I think it is possible, it is really possible that we can build effective treatment strategies for upper motor neuron diseases. So that's why I want to share this with you today, and we can discuss. But I think we are getting very close to touch the finish line, and that's why, as Norma said, we are entering a different phase. Like initially in the first phase, it was no generation of knowledge, because there was a huge lack of knowledge, right? Now we have knowledge, and of course we will need to develop better knowledge, more detailed knowledge as well. And we have tools, reagents, we have model systems, we understand the genetics. So we understand, but how do we build translational research so that this knowledge turns into treatment? Okay, so that's what I would like to discuss with you today, and maybe summarize what our lab has been doing. So of course, feel free to ask any question you want. But I think, let me go one more. So we had numerous challenges along the way, right? And the challenge was that the cortex is very heterogeneous, and there's selective vulnerability is that these upper motor neurons, here labeled in Ramon y Cajal's drawing more than 100 years ago, these large pyramidal neurons degenerate very specifically or selectively and early in upper motor neuron disease patients, while other neurons in the brain remain relatively healthy. So why is that, right? Why is that? And these neurons are very few in numbers. If you look at the whole cortex, they maybe make less than 0.01%, right? So then if the numbers count, they are not so significant. But if the function counts, they are very significant. Because even though their numbers are very small, what they do is second to none. So they actually initiate voluntary movement. They modulate voluntary movement. They project all the way to the spinal cord, and they give an order to the spinal motor neuron to do, to execute function. So it's not really the numbers, it's really the function, right? And then I consider them similar to like the CEO of a company. That you have one CEO of a company, but you have thousands of employees, right? So if you think about the numbers of CEOs, they are very few, but they execute orders, or they modulate, or they actually have a big impact on the company, right? So the upper motor neurons may be few in numbers, but they are very important. And but the tools that we have usually focus on the numbers. They say, oh, there are very few. How are we going to see them? How are we going to isolate them? It's not easy to isolate them. It's not easy to see them. So many people ignore them. And because the brain is so complex, the upper motor neuron was ignored for a very, very long time. And the other challenge is the heterogeneity of the disease. Like, even though this is a rare disease, right, it doesn't affect millions of people, it affects a small population of people, yet within that small population, there is a huge variation. So patients, even though this is a rare disease, the underlying causes of the disease could be many. So some can have, let's say, TDP pathologies, some may have oxidative stress, some lipid homeostasis, axonal transport defects, it could be many. So then it's also harder, much harder for drug companies to be interested, because then it's not like one drug is going to be a solution for everybody. Even though this is a small population, now they have to think about the variation among the small population. So then drug companies are also less interested, right? So there are many, many challenges. And once we know the challenge, we can actually start building solutions. But one thing that we realize, I mean, everyone in the fields now realize that these neurons are critically important. They are very important for the initiation and modulation of movement. So if there is any neuron degeneration, neuronal vulnerability, of the upper motor neurons, then it directly affects people's ability to walk, to talk, all the voluntary movement is primarily affected. So even though their numbers are very small, their job is huge, and they are very significant. And now, we can't ignore them. We definitely cannot ignore them. But the thing is, also, remember I told you the underlying mechanisms are different, but it's not only the mechanism, it's also, if you look at the mutations, there are so many different mutations, right? SPG4, SPG7, SPG this, SPG that. There are numerous mutations. And if you look at the genes, they all code for a protein that is not really related. Those proteins do different things. So then you wonder, how come a protein which does this gets mutated, and then you have HSP or PLS, and then a mutation that codes for a protein which does something totally different, and then again you get HSP PLS. I mean, why? And why do they degenerate? What is the mechanism? How can we tell these patients apart? How can we have better inclusion criteria? How can we understand how best to treat each patient? So those were the knowledge gaps, right? And then for the translation, I mean, how are you going to translate? Are you going to use mouse models? But then we realized mouse survival is not equal to patient survival. So then how do we translate? Like, you can do experiments with anything you want. You can get data, anything you want. But then how translational is your data? How is translational is your knowledge? That's the key question. And again, there was many limiting factors in terms of translation. So we had numerous problems. And of course, for the technology, all right, so how are we going to develop a drug, and how are we going to make drug companies interested for a rare disease that develops due to many heterogeneous underlying causes? There's no biomarker, there's no gene therapy, technologies are lacking behind. And then it was with no touch zone, like no-go zone, like, no one wanted to deal with this, right? Because you don't have the knowledge. There's questions about translation. There's lack of trans technology. This is what we were 10, 15 years ago. So then it was almost like, okay, we don't want to touch that. You don't want to do that, because it's impossible for any drug company or, any translational effort to be effective, right? So that's how the landscape was when I started Northwestern, because again, no model systems, nothing. Like there was really nothing. And people were telling me, why do you study upper motor neurons? Who cares about upper motor neurons? Oh my God. So those were the tough days, right? But then I'm a bit stubborn, which is a good thing sometimes. And then I say, no, these neurons are important, okay? And they initiate movements. Yes, I understand they are very few in numbers, but I actually stood up and I said, okay, we have to study the upper motor neurons. Then, in addition to we also have roadblocks for success, right? And then the roadblocks are, we don't know which patient develops the disease due to which mechanism. Okay, so that's a big roadblock because patient comes to the doctor and they have similar clinical manifestations, but you don't know if they develop the disease due to A, due to B, due to C. Yes, knowing the mutation helps, but usually each patient is a different story. And then let's say you're going to develop a drug. What is the target? What are you going to target in your drug, right? So that's also a little bit of a limiting factor, roadblock. And, what will be the therapy? And how can you tell the drug treatment is working? Again, you need biomarkers, or you need a way to show that the treatment is working, right? And there's no quantitative outcome measure that tells you that upper motor neuron health is improving. So how will the drug companies know if their treatment is good? And plus, very few scientists were working on upper motor neurons, so it was hard to build a team effort, and it was very hard to get NIH grants for financial support because, again, there's not much interest in the upper motor neuron diseases. There's no special call. There has not been a single special call from NIH for the upper motor neuron diseases. Can you imagine? Now, the cut pay line for Alzheimer's disease, 25%, sometimes 30%. They almost give like money away, right? If you're working on Alzheimer's. But when it comes to upper motor neuron diseases, it's maybe 14% at best. And there's never a special call for upper motor neuron diseases. Okay, so it's not in their radar. And it's not like a hot topic, or, that's bad. That's also sad. And so those are some of the roadblocks. And, we have to face that. Like, now this is as black as it gets, okay? From now on, it's going to get brighter, I think. So, but defining the problem is very important, because if you don't know what the problem is, you cannot begin to develop solutions, right? So as long as you define the problem, you understand the problem, you understand the roadblocks, limitations, this, then we can begin to solve them. And I think that we have taken a very important step. For example, you remember I told you that patients have different mutations, and how come different mutations cause the same disease? So we published a paper in Scientific Reports for ALS, but we also published a paper for HSP and PLS, okay? And I'm gonna tell you what I mean by this. So, so far, there are some genes that are causative of HSP, or associated with HSP, or HSP linked, or genes that when you have mutations, it leads to PLS, okay? So these are all the genes that we know so far. And they all code for a different protein, okay? So then we thought, okay, imagine that each of these genes that code for a protein, right? That particular protein must be so important for the upper motor neuron function that when you have a mutation and the protein cannot do what it was supposed to do, then the upper motor neuron says, you know what, I can't do this, I can't. And then they go degeneration. So then this protein, so you define function by the interaction domains. Like each one of us, when we are alone, right, in an island, we cannot be effective. So for us to be effective, we work in a company, we work with our friends, we communicate with each other. Like one-man island is ineffective, right? So when we start to get together, then we create some value, correct? So proteins are the same. So single protein cannot do much, but when proteins interact with each other, they do this, they activate this, they inhibit this, blah, 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 they start this reaction, they start that reaction, blah, 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 and then something happens. So then each of these proteins are important for something. So in some patients, it may be the, I don't know, SCL16A2, it may be SPG20, it may be this, but it's a different function. So then we say, all right, how do we find the function? We look at the proteins and their binding partners and say, all right, all these binding partners, what do you do, right? And why what you do is so important. Then we look at the upstream regulators, cellular functions, and so forth. We begin to realize what is really important for these upper motor neurons. And then he says that lipid homeostasis is very important. Cytoarchitectural dynamic is very important, that they have to maintain the axon structure. They have to maintain their apical dendrite. They have to maintain the cell structure. And the upper motor neurons are so unique that they are located in the brain, but their axon goes all the way to the spinal cord, and their apical dendrite goes to the top of the brain. So they are elongated, they are very fragile, and yet very strong. So they are fragile, but very strong. And they need to make sure that their walls are well built, that they are stable, they can stand, right? So that's why the cytoarchitectural dynamics are very important. And then they should also be strong against the neuroimmune modulation. Like when astrocytes, microglia, all this come, they need to be able to protect themselves. So we realize that those are the key themes that are important for them. Okay. So these are to show some of them, but then now, based on this mutation information, we begin to realize why they degenerate. What makes them upset? What makes them angry? What makes them, say, all right, I'm done, right? So the moment we understand why they are upset, then maybe we can begin to make them happy, okay? So then we're trying to solve their problems. They have many problems. They have ER stress, which means, they cannot make proteins. And the ER says, you know what, I'm not making the protein I want to make, and these are not folding properly. I can't iron them. I can't transport them. Mitochondria says, I'm not making enough ATP. We don't have energy. And then proteins aggregate. Nobody's taking the trash out. Nobody's bringing the money to house. They don't have funds. They don't have cleaning system. They don't have proteins made. And then the transport is lacking. Axon's transport is not going. And they are not able to breathe, right? So we understand their frustration. Okay, so once we understand their frustration, now we just have to make them happy. So now you remember one of the question was, what is the translation? Where is the translation? All right, so we realize the translation is not at the species level, which means human is human, mouse is mouse. If mouse lives long, that doesn't mean humans are going to live long, right? So previously, all the drug discovery studies, they give the drugs to the mouse, and then if the mouse lived long, they said, oh, wonderful, the patient is going to live long. And after 35 trial failures, they realize, no, that's not true. Mouse can live, but human will not. So then, go one level below. Let's go from species to cells. The upper motor neurons in a human and the upper motor neuron in a well-characterized mouse model at a cellular level looks pretty much the same. Large pyramidal cell body, long apical dendrite going to the cell top layers of the brain, a long axon going towards the spinal cord. They are excited to the neurons. They are born pretty much the same time. They migrate pretty much the same. At a cellular level, there are many resemblances, okay, at a cellular level. But let's go even one level deep. If you look inside them using electron microscopy, for example, you look at their mitochondria, you look at their ER, you look at their Golgi, you look at their, all the organelles inside the cell, they are identical. So a mitochondria in an upper motor neuron of a human and the mitochondria of the upper motor neuron in the mouse are identical. So if I were to give you a slide and show you, okay, tell me which one is which, you wouldn't be able to tell. Then we look at a patient, okay, and the upper motor neuron of a patient, their mitochondria, and we realize, oh my God, it's all broken. They don't have inner mitochondrial membrane. Mitochondria may be swollen. So there are many mitochondrial problems. Then we say, How about the mitochondria in the upper motor neurons of the well-characterized mouse models? Oh my God, it's the same problem. Inner mitochondrial membrane is broken, they are swollen, mitochondria has problems. So then the translation, okay, so the common denominator is at the cellular level. And if you look inside the cell, the pathology is the same. So ER stress is the same, mitochondrial defects are the same, lipid problems are the same, nuclear membrane defects are the same, cytoarchitectural problems are the same. So then the cell, the neuron, speaks to us. So the translation is not at the species level, the translation is at the cellular level. So that was a remarkable discovery, because now we can actually begin to ask questions, and we can begin to receive some answers. All right, so now I'm going to stop here and ask you, if you have any questions for the section that I have spoken about. Are there any questions? I see one.
Okay, folks, you hear her calling for questions. So hit your reaction.
Yeah. So during the talk, you can continue writing in the chat, and I will stop at at some points, and then I can read the chat. So here there is have mutations in a variety of genes. How does degeneration of upper motor neurons affect proteins made by various genes? Oh, this is an excellent question. All right. So then, when there's a mutation in a gene, okay, let me tell you this first. Genes code for a protein, right? Okay, so that's an information that we know. And when there's a mutation in a gene, then the protein has a different amino acids, or has a different, if it's, let's say, expansion, there are different mutations, right? One could be a single nucleotide mutation. So then there may be a change in the amino acids. But sometimes mutations could be such that there's expansion in the gene, so that now you have a very different protein. Like, it's either longer, or maybe there could be a deletion in the gene, so that now the protein could be shorter, right? So depending on the mutation, the protein may be different. So now the location of the mutation is also very important, because the protein has multiple domains. With one domain, it can be like a kinase, right? Let's say it adds a phosphate group, it removes a phosphate group. Let's say it activates ATP, it reduces activation. It has multiple groups. Or in one, it binds RNA. In the other domain, it binds a protein. So different domains can do different things, okay? So then if there's a mutation in one domain, then the protein may not do what it is supposed to do with that domain, but the other domains can still be a little bit functional, okay? So the mutation is important, but the location of the mutation is also important. So then when there's a mutation in a gene, like for example, some patients, they have mutations in the same gene, okay? But some patients are very severe. Oh my God, very severe. And some patients, not so much. It may all depend where the mutation is. The severe phenotype patients may actually have the mutation right at the site where that domain is critically important for its function, okay? But for other patients, the mutation may be at a site where it's not so much important, okay? So the protein may still, maybe it's not functioning 100%, but maybe it's functioning 60%, right? So the location of the mutation is also important for the protein-protein interactions and for the function of the protein. All right, so I see two hands.
Thank you, Dr. Ozdinler. I appreciate that. Now that was Mary. Mary, do you have another question? If you do, you're on mute. Go right ahead with your question.
Yeah, hi, doctor. This is Ed Duffy, and I was curious about what took place in Canada this past month with that little boy.
I was thinking about that.
Is there, I believe that there was some gene editing that went on. I'm not sure, but I was wondering how that would translate to a larger population of gene mutations if it works.
This is for the gene therapy, right?
Yes.
All right,
Yes.
so we're going to come to that.
Okay.
I will have a section for gene therapy, yes. Yeah, so we will discuss this. Yeah, hold your question. Okay,
Okay.
I will answer that. Any other questions? These common problems, then tell us the translation is at the cellular level. So now, you know, some people try to use iPSC cells. So we don't use iPSC cells in our lab because they think the translation is at the species level. So they use, they take, let's say, fibroblasts from human, and they, you know, change it or differentiate to motor neurons, right? So then it's the same species, okay? And then they force these cells to differentiate in a dish, okay? But then for them, the translation is at the species level. But for us, the translation is at the cellular level. So then, despite that the one is in the mouse, the other one is in the human, if you take the upper motor neurons as a pure neuron population, because they are born, they differentiated, they developed, they are integrated into circuitry, they are like upper motor neurons here, upper motor neurons there, right? So they resemble each other at the cellular level. So then we actually think that the cell has more power and more translational impact. So then we made the reporter line to be able to see them, to be able to differentiate them so that we can actually isolate them. So this was very important because for the first time, we made a reporter line that allowed us to see and to distinguish upper motor neurons from all other cells and neurons in the brain, okay? So after this publication, you know, we got NIH grants, we got awards, this, that, because this was actually the first reporter line which was cell type specific. There are many reporter lines, they, you know, they label this and that, but this was very selective to upper motor neurons in the motor cortex. So now that's very important because now we shift our focus from mouse to human, let's say, translation to the cells. And we confirm that these are upper motor neurons. I'm not going to go detail with labeling, with molecular marker expression, with electrophysiology, this. And, you know, we got many awards, blah, blah, blah. So now we actually have the first upper motor neuron reporter line. And that was a game changer because now that you can see, you can modulate. Now that you can see, you can isolate. Now that you can see, you can ask questions, right? And then we focused our shift from mice to neurons. And now we begin to ask upper motor neurons, you know, why do they degenerate? Why do you degenerate? So that we learn directly from them. So now I became an upper motor neuron translator. Upper motor neurons begin to speak to me now. And we begin to have a conversation with them, right? And to have that conversation, we use many omics. We use proteomics, lipidomics, metabolomics, all these omics, RNA-seq, you know, we use many technologies, and those technologies are also developing as we speak. But again, 15 years ago, we didn't have those technologies. So technologies are also up and coming, right? So we can ask them which compound they like. I mean, they speak to us, and I love talking to my upper motor neurons. So now we cross these reporter lines with many of the well-characterized disease models that show progressive upper motor neuron loss. SOD was one. Our group actually was the one who showed it. TDP is another one. TDP pathology is also seen in HSD-PLS patients. Alsin, for example, is also seen. Now recently also spastin. So all these mouse models are generated by the mutations that are detected in upper motor neuron disease patients. So they mimic human. So even though they're mouse, they're actually part human because we make them express the human mutation. So we humanize the mouse, right? And then now that we cross them with the reporter line, now we have diseased upper motor neurons that are also fluorescent, okay? So now that they're fluorescent, this is very important because they shine. They are GFP. They are like, you know, it's beautiful because they are bright, okay? So we use fluorescence activated cell sorting. And then we say, all right, you come over here, all right? I don't want all other, you know, all other neurons. I just want you. So we actually use fluorescence activated cell sorting, and then we isolate only the neurons that are diseased, only the neurons that are vulnerable, not other neurons, just those that are diseased. Then we do all the omics. RNA-seq, proteomics, metabolomics, lipidomics, right? And we ask them, tell me, what makes you upset? He says, oh, I express this gene, I can't express that gene, that protein is not interacting with me, my ion channel is not spliced correctly, I can't carry the axon transport, my proteins are, oh my God, they have all these problems. And then I say, all right, so the alsin upper motor neuron, tell me, what is your problem? It says, oh, my lipids are not working very well, and this is not going to, oh, alsin has a different problem, profilin has a different problem, spastin has a different problem, they all have problems. And some of the problems are common, some of the problems are unique. And one of them, for example, says, oh, the axon, because the axon is a very long I-90, or, it's the interstate, right? And then there are holes on the interstate, but the holes are so big that the trucks fell. I mean, trucks go down, so they can't really have the trucks going up and down. And I need this shipment. It's not coming, If I don't have that shipment, I can't make another one. And I don't have enough money to pay another shipment. That must be coming. Where's FedEx? I mean, where's the road? Roads are broken. But I need that. It's not coming. It's, oh my God, so the roads are broken. So that's the problem. Roads are broken. Okay, fine. The other one says, oh, my lipids. Oh, no, I need this sugar. I need that. And they're not giving my sugar daddy. Where's my daddy? Where's my sugar? The sugar. Says, oh, yeah, the roads are working, but I'm not getting enough sugar. I said, oh my God, so the sugar is a big problem for one, the other one. I said, oh, they all have different problems. The good thing is they begin to speak to us, so we know what their problems are. And they are all unique problems, and there are some common problems, okay? So when they speak to me, I really like it. So then the other thing is they tell different problems along the way. For example, when they're a teenager, it's a different problem. When they're a young adult, it's a different problem. When they just got married, it's a different problem. When they are 50 years old, it's a different problem. So they all have different problems at different time points, okay? So it depends also which state of the disease you are at. The problems may be different. So then we also do longitudinal studies to see which problem occurred first. And if I solve that problem, would I be able to eliminate other problems? Or are all these problems independent of each other? So I need to, solve multiple problems at the same time. Which problem must I solve first? Which problem must I solve at the same time? Now we are in the the problem solving stage. So the proteomics that we use is very important as well, because that also tells us which proteins they interact, which proteins they don't interact, and how that changes. So there are some examples. For example, these are upper motor neurons that are diseased due to SOD1 mutation at P15. So this is the time mouse are this big, their eyes are closed, they're still with their mothers, but even at that stage, at a cellular level, even though there's no disease, nothing, right? But at a cellular level, the cell says, oh, my lipid metabolism, I have a problem with the lipid metabolism, transport, I can't transport. So the problems begin to emerge very, very, very early. So then by asking the cell, we also begin to understand what is the early, the first problems that you have. And then say, oh, I think it's the mitochondria. See, it's the mitochondria. And it says, I think I'm having a neurological disease, this is like P30. They just became adults, but still no symptom. It says, I think I'm having a neurological disease. And I think there's some injury, abnormal. There's something wrong, okay? And this is the active time of gene expression, trying to overcome. And then my morphology, like cytoarchitectural dynamics, I can't keep my morphology, I can't keep my walls. And the cell-cell interaction, my spines are losing, I can't really have good interaction. So the signaling, the communication, I'm having some problems, but you can't really tell, what the problems. And it says P60, oh, again, the polarization of mitochondrial membrane, I can't make enough energy and movement, and oh, it's this and that. And P60, now I begin to show symptoms. P90, it says acute phase response signaling, like come clear me, leukocyte extravagation. So it's like that receptor signaling, I'm dead, like doesn't even bother. Like it now at this time, P90 is like towards the end. So now the cell says, I can't really make this. Okay. So now we have to begin to help the cell before they reach that state. Okay. But even at P90, they are still alive. You believe it or not. So they are not cleared. The cells are still there, but they are very pissed. Like they are so unhappy. Okay. Then I think we have to find ways to make them happy. So now with all these omic this study, we understand why they degenerate and what is their major problem. Again, protein aggregation, endoplasmic reticulum problems, mitochondria problem, upstream problem, and cytoarchitectural problems and lipid problems. So these are the major problems. So if we solve these problems, we would be able to help most of the upper neuron disease patients. Okay. So now we understand what is the target. So now we also understand the targets. We also understand the problem. So now, you know, it's just going into biomarkers, because now we need to find a biomarker that would tell us that the application is working, that we can actually see that it is working, and so forth. Beautiful thing is, even though you may think, oh, mice is mice, blah, 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 but at a genetic level, can you imagine that we are about 95, 96% homologous? Like we share so many genes. And there are some proteins, especially if they are key proteins, very important proteins. Their function is so important. They're 99% homology, 96, 97, whatever. They're like almost identical. So then when you do all these omic studies, okay, and then you see some proteins going up over time or going down over time. And if you are lucky, some of those proteins are secreted to the blood. So they're in the serum or they're in the plasma. So then there are also patients who have mutations similar to the mouse models that we have, right? And you can collect serum and plasma from those patients. And now that you identify clinical proteins that go up or that go down, then you can ask the human, do you have the same protein in your blood? Yeah, do you have it? Is it in the serum? Is it in the plasma? What is the level? Does it increase with disease? Does it decrease with disease? And that is your answer for a biomarker. So then if you actually find a protein that goes up with the disease or down with the disease, and it correlates to the disease progression, the rate of disease progression, that's the biomarker that we all need. So now we actually have a huge understanding of all the proteins that may be telling us something. Now we actually got the DoD grants to look for the upper motor neuron biomarkers for ALS with our colleagues at Harvard. So that's a 2-year grant. And by the way, we got 2 DoD grants all at the same time. That's $1.5 million. And they tell me that it's very few people, you know, get 2 grants all at the same time. So I'm very happy that we got those 2 grants. For the biomarker study, now we're going to see if we can really identify biomarkers that would tell us about the timing and extent of upper motor neuron loss. Because if we do, then drug companies will show more interest, because they will know if application of their drug has an implication or not, right? Because when they give the drug, they want to know, did it work? Then we'll say, oh, let's look at the biomarker, because if actually upper motor neurons degenerate, that biomarker should go up. Oh, it's not going up, so it must be working. Or normally it goes down. Oh, it's not going down, so it must be working. Or it's going down. I don't think it's working. You know, we will be able to tell them if they have success or not. All right, so now we're also moving into the biomarker field. So if we, even though it was an untouched territory, now we begin to realize there may be potential biomarkers, and it's in the serum, it's in the plasma, and we're gonna get them. All right, we're running after them. We're gonna find them. Okay, so that's what we're working on. All right, so now these are also some of the biomarkers that we identified that go up or down, and now we are looking at them with more patients. And ALS patients actually there are more serum and plasma samples, but when it comes to HSP-PLS patients, we don't really have too much blood collection for the serum or plasma. So it would be really nice that patients donate their blood so that better biomarkers can be detected, not just for ALS patients, but also for HSP and PLS patients. All right, so I'm going to stop here and see if there are any questions for the section, for this section. And I see some in the chat. I would like to know if there's any blood test to identify misfolded protein. How do lipids in the cells relate to what the average person knows about lipids? Okay, so if there is any blood test to identify misfolded protein, I don't know if that will be a correct answer that I tell you, but let's say in the TDP pathology, right, there are misfolded proteins, protein aggregations, right? And the TDP actually is a protein that splices messenger RNA. But when there's a mutation in TDP, the splicing doesn't work very well. So we already know which genes TDP splices, like stathmin 1 or stathmin 2A is one of them. And if the TDP is not working well, so you have a cryptic messenger RNA, which leads to a cryptic protein. So if you can detect that cryptic protein in the blood, that will tell you that the TDP is not doing its job. So that there's a good chance that there's TDP pathology. So there are some proteins, especially for TDP, because we know it is, working on messenger RNA. Stathmin is a good example. I think UNC13 or others are a good example, but it's not just those, like many are coming up. And so the cryptic protein is important. That's why, remember I showed you the proteomics. It's not only the bottom up, but sometimes you need to do top down, which reads the whole protein so that you can actually see if there are any splice variations, if there are any post-translational modifications, because those proteins would be very informative. Okay, so now, how do the lipids in cells, relate to what the average person knows about lipids? I don't think it relates. So how do lipids in cells relate? Maybe I don't understand the question. What the average person knows about lipids?
I'm just kind of confused about how we know there's cholesterol and that kind of thing. That's kind of like an issue with lipids. I didn't know how that relates to the lipids you're talking about in the cell.
Okay, so the lipids are very important. Cholesterol, you gave it a good example, but lipids are also the they make the membrane structures. And inside the cell, there are many membranes: mitochondrial membrane, outside membrane, inside membrane, ER membrane, Golgi membrane, Golgi stack 1, stack 2, stack 3, all these membranes, cell wall huge, axon. Imagine the axon. Imagine all the lipids, because it's the cell wall is by lipid layer. So lipids are like, let's say, there's one round, and then there are two legs, okay? And the legs are, they don't like water, so they want to stick to each other, so they're inside, and then that makes a bilayer. So lipids, if you don't have enough lipids inside the cell, become leaky, the cell cannot maintain its organelles, membranes. So, and especially for an axon, for an upper motor neuron, they have a long axon. The health of mitochondria is very important. The health of ER is very important. So they are very much dependent on production of lipids, or modulation of lipids, or the lipid homeostasis. They are very dependent on it. So I don't know if that answers the question.
Thank you.
So how can we donate blood samples for your research? That's a very good question. Let me not answer this now. Okay, so last time I answered, I got into trouble. So we'll talk another time. Okay.
Yeah, this is a little bit different than the question I had in the chat. We all have mutations in genes, but our HSP or PLS progresses over time. So is the progression of the disease because of further degeneration in upper motor neurons, or is it because of change in the mutations of the gene, the way they express proteins?
Actually, the mutations in the gene do not change over time. So whatever mutation you have that mutation. Okay. So it progresses, let's say the disease progresses for multiple reasons. One of them is, let's say, whatever the gene was mutated, that protein was critically important. And now the cell cannot make that protein. So it says, I wish I needed HSP7 now, that protein, I really need it now. And I don't have it. okay, maybe I'll use this instead, but it's not doing what I really need that. And then it says, I definitely need that. I mean, I just need this. And so then the stress becomes even more like you really need that protein, and then you can't within its absence, you cannot do what you were supposed to do. And then you begin to suffer. Then you say, all right, I just can't do it without this protein. Okay. And then the neuron becomes stressful. But then, as I said, like no neuron is an island. So let's say your neighbor is very stressed, okay, and your neighbor's it affects you as well, okay. So then you have an angry or sad or not functioning neuron, and you are supposed to write a grant with them. You won't be able to write a good grant with them. Or you are supposed to go on a vacation with them. They're not going to be a good camper with you. So then you begin to feel the stress as well, right? So the neurons interact with each other, and the mutated protein interacts with other proteins. And when someone in your community is very upset, after a while the community becomes very upset, right? So then the problem does not dissolve, the problem accumulates. So the problem just gets worse and worse and worse. So I think that's why the disease progresses, because it may initiate at one point, but now at that point, now it's just like, it's in the kitchen, now it's in the house, now it's on the street, now it's on the neighborhood, now it's on the city, now it's on the, I don't know, it's just, it's, but then you say, oh, it all started in the kitchen. Oh, now it's the whole city on fire, So then I think as the problem is not solved, it's like the, what do you call it? It's the snowball, right? So it gets worse. And I think we need to identify the problem as early as possible and solve it as early as possible. I told many things, right? And now biomarkers will be very important, because then if you look at the patients, we'll say, you're developing the disease due to this, or you're developing the disease due to that, due to this, due to that. So we will know what is the major problem for each patient. Then the good thing is there are drugs developed for these specific targets, okay? So that's also the development in the drug discovery field, because it's mechanism focused, okay? So it's not the disease focused, it's the mechanism focused. So then we can actually, rather than including everyone in the clinical trial and then fail every clinical trial, we can actually say, all right, we're going to be very smart with whom we include in this clinical trial, and we will actually have effective results, right? So we can increase the success rate of clinical trials. So that's also very important. Okay, good. So now let's get back to our table. Remember the lack of knowledge, why motor neurons degenerate. So now we understand. And why so different mutations with similar outcomes. So now we begin to understand. And now lack of translation. Now we know it's the cellular level. So now we understand. And what is the target? Now we understand, right? Because these are the targets. So see, we are making progress. Okay, that's good. And, we make this mouse model, we can see them, perfect. And we mated them with different disease models. We made disease models, perfect. So now we can tackle drug discovery. Because beforehand, there was no drug discovery for upper motor neurons, because they didn't even see the upper motor neurons. They didn't even know the upper motor neurons, right? So now we can see them. Then we said, all right, motor neurons, you tell us what you like. You tell us if you like this drug or not. So now we can actually begin forward for clinical trials for the upper motor neuron diseases, and we ask the motor neurons. And now we also incorporate upper motor neuron health, right? We developed this novel screening platform because we have disease models. They have GFP upper motor neurons. We can give them different drugs, either orally, injection in the water, or this and that. Then, because we know by time the upper motor neurons progressively degenerate, let's say this is early, this is late, they degenerated, and then we can ask them, upper motor neurons, are you happy? Because if they are happy, why would they degenerate? So that you would retain them in the upper motor neuron, right? In the motor cortex. So most importantly, you can also put them in culture. Can you imagine? You dissociate the motor cortex, you put them in the culture among all the other neurons. The upper motor neuron says, hey, I'm here, because it's GFP. because it says fluorescence, so you can see the upper motor neuron among all other cells in the brain. Then you add the compound of interest and you ask the upper motor neuron, hey, do you like it? And if it likes it, it's going to send a long axon, it's going to branch, arborize, I'm loving it. Or if it doesn't like it, then it's going to shrink, it's going to degenerate. So they begin to speak to you. They begin to tell you what they like. They begin to tell you what they don't like. And, there's a communication with the diseased upper motor neurons. So now you also have upper motor neurons that are diseased due to, let's say, SPG4, or that are diseased due to alsin, or that are diseased due to this mutation, that mutation, different mutations, right? And you remember, they diseased due to different reasons. So there may be one drug that one person likes, but the other upper motor neuron may not like that one. It says, you know what, I can tolerate it, but it's not my favorite. Or the other one says, oh, I love it. And they may have different drugs that they like. So you can even tell with the drug companies, well, this drug is actually pretty good for a patient with this mutation, but not so good for patients with that mutation. So they may even have inclusion criteria built based on the mechanism that these patients develop the disease, okay? It looks really nice. You can see them, GFP, all these are other neurons. You can see the astrocytes next to them. And I like this because look at the upper motor neuron here. There's an astrocyte attached to its back and it's trying to run away, right? You can even see their interactions with astrocytes, if they like it or if they don't like it and so forth. So, you begin to become friends with them. So I need to tell you that when we were developing all these drug discovery platforms and everything, Dr. Silverman, independently from us, was generating numerous compounds. And Dr. Silverman is a very well-known medicinal chemist. He's the inventor of Lyrica, and he made the compound NU9. And we met at one of the ALS Association meetings in Chicago, and we became friends. And I told him, do you know if NU9 works in upper motor neurons? Because NU9 was developed to reduce protein aggregation of SOD1. We said, you know what? We actually have the system. Let's test it on our SOD1 model. So we gave them gavage over time, many different doses and so forth. And we published this paper. It's actually a very seminal paper because it tackles two important questions in the drug discovery field. One of them is we try to develop drugs not for a disease, but for a mechanism. Plus, we actually ask the neuron whether they like it or not. So it's cell type specific, okay? So we don't care if the fibroblasts like it or not, right? We ask if the upper motor neuron likes it or not. And we also look for targets. You remember I showed you mitochondria, ER, so we have very good targets. And we ask whether at a mechanistic level, we have some improvement. I don't know if I should show this, but they made it, it was very, I'm not going to show, but this was actually very nice because when we published in the Wiley Publishing Group, they said this publication is so good that we're going to make a movie out of it. So they made a movie out of our paper. I'm not going to show this movie now. It's basically showing that it's cell-based and mechanism-focused drug discovery. Okay, so NU9 is actually the first compound that improves the health of diseased neurons. So our group identified not only the first reporter line, but also the first compound that improves the health of the upper motor neurons in less than 8 years. And if we look at the progress of research, that's amazing. That's very fast. So how were we able to do this? Because we knew targets, right? And if you look at the mitochondria is all falling apart, ER is falling apart. So now we ask a question. When we give NU9, can we improve the integrity of mitochondria? Can we improve the integrity of endoplasmic reticulum? And when we do, because that's the major problem, when we do, would that translate to improvement of health? So we go down, 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 all the way to the cell, to the organelle, and then up, 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 up to the cell body, to the circuitry, okay? Yes, that was the case. You can actually improve the health of the mitochondria. Look at this, it almost becomes to the wild type levels. You improve the total number of the mitochondria, and so mitochondria is improved. And then you look at the Cell numbers, yes. Look at this. When you give 100 milligram per kilogram, there's no more degeneration. So they retain in the motor cortex. They say, you know what? You solved all my problems. Why do I degenerate? Right? I told you I can't make ATP and mitochondria is a problem. You solved it. And I told you my ER is really bad and I have ER stress, I have this, and you solved it. Okay, so why do I degenerate? Right? So we can switch the cellular degeneration path to, I don't need to die. I'm fine. I'm fine. No, thank you so much for solving my problem. I'm gonna hang out. Okay, I'll be here. I'll be okay. And they retain. They retain in the motor cortex, and that's what we want, right? So it almost becomes to the wild type levels. That for 60-day treatment, normally they would definitely go progressive degeneration, and after 60 days, they say, you know what? I'm cool. I'm here. And that was amazing. That was just like, oh my God, like nobody has ever seen like this before. And then we looked at the apical dendrites. Okay, can we really restore the apical dendrites? And yes, we were able to see many more apical dendrites. They were intact. So that's another good reason to believe that they are not just staying their cell body, but they most likely will have better inclusion to the circuitry, right? Because now other neurons may be able to speak to them. Because if you have an apical dendrite like this, nobody speaks to you. You don't have spines. You don't have. I mean, how can you talk to a neuron that has an apical dendrite like this? So now we improved the apical dendrites. Then we said, okay, now that was a pivotal moment because NU9 was found to reduce protein aggregation in the SOD1. Okay, and yes, now when we give NU9 to the SOD1 mouse, we solve the problem. Okay. But how about TDP? Because TDP and SOD1 are two, like apples and oranges. They are different proteins. It's a different aggregation. It's a different strategy. And even the patients with SOD1 mutation, they don't really have TDP pathology in their brain. So this TDP and SOD1, they are a bit separate, okay? They are like different. So if NU9 can reduce protein aggregation, SOD1 protein aggregation, can it also help TDP, which is a total different biology, which is a total different protein aggregation, but the theme is different. It's protein aggregation. And the other thing is, in the TDP mouse, okay, so we made the mouse, oh my God, like I'm showing you a figure here, this picture, right? Making that figure, it took us 5 years. Can you imagine? One publication, one figure, it took us 5 years because we generated the mice, we did all these behavior essays, we retrogradely labeled them, we counted cells. So this is 5 years worth of work in one figure. But we made them. And the mouse also behaves like the human with the TDP pathology. And then we asked the mouse with the TDP pathology, which is also seen in HSP-PLS patients. If I give you NU9, would you like it? And they said, oh, I love it. My apical dendrites are more straight and I don't have as much degeneration. I'm happier. I'm actually doing much better. I'm doing much better. And I said, really? Like, why? And then they said, yes, my mitochondria is better. My ER is better. So then we realized that NU9 is not a compound just for SOD. It's also a compound for TDP. Then we're talking about a broad spectrum of patients. ALS, FTLD, HSP, PLS, all right? So then NU9 was important for the upper motor neurons. In the SOD1 mouse, we asked spinal motor neurons, do you like NU9? They said, we don't care much, not so much. So then we realized, oh my gosh, so the spinal motor neurons and upper motor neurons, we think that they are the same neurons. One live in the top floor and the other one live in the bottom floor, but they're all motor neurons, right? No, they aren't. They are different cells. They are different neurons. And then we realized like this is the first compound that the upper motor neurons prefer, they like, but not so much the spinal motor neurons. So then this also tells us something very important about drug discovery, especially for the ALS, because can you imagine in ALS, all the drugs that they tried in ALS patients, I don't know, 40, 45 so far,
Mm.
none of them have ever asked whether that compound also improves the health of the upper motor neuron. So if it improved the health of the spinal motor neuron, they said, oh, fine, spinal motor neurons, they like it. Or, they never asked upper motor neurons. So it is possible that some drugs spinal motor neurons liked, but upper motor neurons didn't. And if upper motor neurons didn't like it or they didn't care for it, they continued degeneration. And if our upper motor neurons continued degeneration, then the cortex connection from the brain to the spinal cord may be lost. So you need to improve both of them in ALS. That's what makes ALS so challenging. But for HSP-PLS, the main focus is the upper motor neuron. So you don't really need to worry too much about the spinal motor neurons because it's mainly the upper motor neurons. So that's why NU9 was so specific and so unique, because it had a predominant effect on the upper motor neurons. Okay, so that was very good. Then we said, okay, NU9, and we published this in vivo, right? And we, of course, were curious, like, is it better than the FDA-approved drugs? Like, now there were two, right? Riluzole and edaravone, right? And now we can actually put them in culture, right? And in culture, we can see if they're axon outgrowth, arborization, and this and that. We can see them, right? Here's the upper motor neuron. How beautiful. Look at the axon. And you look at the cell body, boom, it expresses, let's say, the molecular markers, CTIP2. Other neurons, they don't. And then in the wild type, there's, let's say, they have about 300 micrometers in 3 days, but SOD1 is about 200. So there's a big difference. And, they're shorter and they don't arborize. So we know the baseline, right? What is the disease? What is not? Then we add NU9. Then we add Riluzole. Then we add edaravone. Then we look at them and we say, tell me which one you like better. Do you like Riluzole better? Do you like edaravone? Which one you like better? And then if they like it better, then they arborize more. Look at this, look at this neuron, for example. Ooh, we're setting a party, right? The others are okay. So if they're happy, you can tell they're happy. Then we said, okay, we looked, I think, more than, I don't know, hundreds of neurons. And then we said, all right, so it seemed as if that NU9 was making the upper motor neurons much happier than Riluzole and edaravone. Then we said, how if I give them together? Ding, ding. That's again mechanism, because edaravone is a free radical scavenger. Okay, so it takes all the free radicals. The mechanism of action is taking the free radicals. Riluzole is actually acting on the astrocytes. It's making astrocytes less active, or activated astrocytes, right? So that they become less toxic. So the astrocytes help neurons grow better. So then mechanism of action is different. But NU9 mechanism of action is improving mitochondria, improving ER, reducing protein aggregation, improving apical dendrites, helping cytoarchitectural dynamics. So it's 4 things. Like one stone, you hit 4 birds, okay? So that's very unique, and that's different than the Riluzole and edaravone. And guess what? When you add them together, there's also a effect. So then neurons say, wow, I had 1 problem, I had 5 problems, now you're solving them all. Haha, you're getting better at this. And they like the fact that we actually solve multiple problems all at the same time. So if you solve multiple problems at the same time, they will even be better, right? So so now we identify their problems and we're solving their problems. And if there are also, we can also do additive treatments. And that also, it opens the door for personalized medicine. Because every person is different, every person's needs are different, then I think in the future, doctors will say, oh, you have this problem. I mean, your neurons have those problems. Then I think you should take drug A, B, C, D. But another patient, oh, you need to drug C, D, E, F, G or something. Or the other one, G, X, Y. Or the other one, there could be a combination of drugs. And it would be for the mechanism, not for a disease. And that also reminds me to tell you that when FDA was formed in the 1940s, FDA thought that diseases are unique and distinct And Alzheimer's is different than Parkinson's, and Parkinson's is different than ALS, and ALS different than HSP, HSP different than PLS. And then these are distinct diseases, because those diseases were characterized by doctors who observe patients clinically. So the definitions of the diseases were based on clinical manifestations, right? They said, oh, Parkinson's patients shake, Alzheimer's patients can't remember, ALS patients can't walk, but they remember this, that. So, in basic terms, they thought diseases are distinct entities. They defined diseases. And then their goal was to find a drug to eradicate the disease. So they thought one drug is going to cure all. But they did not know cell biology at the time. They did not know proteomics. They did not know lipidomics. They did not know all the omics that we have. Now we realize that these diseases are not distinct. They are actually branches of the same tree. There's the tree of neurodegeneration, and in some branch you have apples, in another branch you have oranges, in another branch you have this, in another branch you have that. But basically there are common aspects of the disease and there are unique aspects of the disease, okay? So then it won't be that one drug is going to cure all ALS patients, or it won't be that one drug is going to cure all Alzheimer's disease patients, right? So there will be multiple drugs based on the mechanism for the patient. So that, of course, is a huge change in our thinking. And that's a huge leap forward. And all the language, everything needs to change. But it has to, because that's the truth, because that's the reality. And now, look, for example, when you add mu9 with edaravone, look at this neuron branching. Look how happy this is. Do you see? Look at this. And metulazole, look at how happy. Do you see? So our goal is to make the neurons happy. Because when the neuron is happy, then the motor neuron circuitry is happy. And when the motor neuron circuitry is happy, then you can actually function. Like when you have motor function, then it makes you walk or talk or dance. Once the neuron is happy, that is the solution. So now we are actually very happy because Dr. Silverman formed the company with his sons, and it's called Akava Therapeutics. And mu9 is actually one of the compounds that is considered for moving into clinical trials. Please check this website. It's Akava Therapeutics. Write it down. Check the website. Read it really well. And pipeline. Learn about this. And I'm also happy that Dr. Hiroshi Mitsumoto is in our scientific advisory board. So I'm the director of the scientific advisory board. And now, for the first time, friends, for the first time, there is a drug out there developing for clinical trials for upper motor neurons. Okay, who's going to applaud? Who's going to applaud? All right, good. So now, you remember, now we are actually making progress in our problem. We've listed all these problems. Now we're making progress. Okay. So this is actually a very important point that I want to mention to you. During COVID, and there was everyone was at their homes and everything. There were two families in Italy, and with their daughters. Daughters are 2 years old and 3 years old, little girls, and they had mutations in their alsin gene, different mutations and different parts of the gene. For example, one of them had it in this RLD domain, the other one is in the VPS9 domain, different domains. All right, so these are the domains of the alsin protein. As you can see, there are different domains color-coded, and this is the three-dimensional structure of the protein showing where the mutations are. And these are the family. This family had the daughter, the mutations. This family had another daughter with different mutations. But they're all mutations in the alsin gene. Mutations are in different places, in different domains. You remember I told you? Okay, but because they have this alsin gene, even though they are 2 years old, 3 years old, guess what? They are ALS patients. They can't walk, they can't get up. And it's not that ALS isn't affecting elderly, even the children it affects, right? So then we said, if we get serum and plasma samples from these little patients, can we begin to identify some key metabolites that would inform us about their disease condition. And so we actually published just the metabolite part, but we have also done the lipidomics and the proteomics, and we haven't published it yet. But I'm just going to show you how revealing that is. That if you look at the serum samples, okay, so there are some metabolites that in the control male, female, none, like there's not even a single, okay? But in patient number 1, AO, and patient number 2, DH, these were very high levels. And the relatives, which mother and father, and they're heterozygous, right? They have half the mutation, and the level was half. And, you know, there were metabolites that were either absent from the control and present in the patient, or very high levels in the control and none in the patient. So now you may say, what does that mean? Okay, I cannot give you the answer to that, so we're still investigating, but there are very key aspects that these metabolites are playing important roles, and their absence or their abundance also tell us what may be going wrong inside the cell. And mitochondria is really an important organelle for the metabolite production and facilitation, you know, of their activity. So when you see all these dysregulation of metabolites, it also tells you that the mitochondria is not very happy. Okay, so then we look at the serum, we look at the plasma. The other thing is, you remember I told you that these AO and DH have mutations in different regions of the protein. Okay, even though it's the same gene mutation, look at that. There are also differences between AO and DH. So it's the same gene, but because the mutation is a different site, there are differences in the metabolite. You know what that tells you? It tells you that if the mutation is here, then this domain is not doing what it is supposed to do, but other domains are doing what they're supposed to do. So if there's a metabolite that is missing in a patient that has mutation here, that metabolite is related to the activity of the protein at that region. So now we even, like, can you imagine the precision here? It's not just the gene now, it's even the location of the mutation with the metabolites. So we were very precise in our analysis. And again, we look at the plasma. There are again some that are either present or totally absent or differentially present in patients, okay? So then we put all these metabolites together and say, metabolites, tell me, what's the problem? What's going wrong? And they say, oh, the carnitine synthesis, we can't really do that too much. And pre-metabolism, methionine metabolism, oh, this metabolism, that metabolism. So there are problems, right? And this was very important to us because then we begin to understand what is the exact problem for each patient. So we were able to, tell or communicate that knowledge to their doctors. And we started the treatment strategy for these small patients, 2-3 years old, because we realized that there were problems with the mitochondria and that mitochondria couldn't do this, that. And then the doctor said, wait a minute, this is actually the same problem that occurs in patients with the mitochondrial this, and there's already an FDA approved drug for this, or there's already this. And because it's already FDA approved, right? You can actually use off-label use, or, there are ways that you can actually try drugs based on mechanism. And now they treat the girls for like 6-8 months. And one of them, the doctor told us, that begin to stand. Can you imagine how happy I was when I got the mail? She was able to stand. So now they treat the girls for like 6-8 months. They collected the blood samples again. Now we're doing post-analysis, like what metabolites are changing pre and post, like before and after treatment. And if we see some of those metabolites change, and does it really relate to the behavioral improvement? So I think if we can understand why these upper motor neurons are upset, there are tools to make them happy. So being able to understand what makes them upset is also with the metabolites, proteomics, lipidomics, and so forth. Okay, so now gene therapy. Remember, there was one question early in the morning. So we can develop drugs for patients as long as they are not ultra rare. If it's an ultra rare and there are, like, say, 2 patients, 3 patients, 5 patients in the whole world, with that mutation, and their case is so unique, and that no drug is going to help them, and it's just ultra rare, right? And then the gene therapy would actually be when I say ALS, I actually mean upper motor neuron diseases, and I use ALS in my examples because we have some of the ALS models with prominent upper motor neuron loss. So then we begin to develop the gene therapy approach. That can we really modulate gene expression selectively in these upper motor neurons? So we published first with retrograde labeling, which means you give the virus to the spinal cord and it goes all the way to the cortex, or then we direct motor cortex injection, because now your goal is to change the gene expression selectively in the upper motor neurons. So I'm actually very happy that we just published in Nature Gene Therapy. We made the cover. There was a news written about our paper, and we actually got the award for, what was it, best read or most read or something article. In any case, but in this paper, we showed that the upper motor neuron degeneration, especially for ALS patients, for the upper motor neuron degeneration is not dependent on the spinal motor neuron degeneration. Because there were so many people thinking, oh, you know what, upper motor neurons degenerate because spinal motor neurons degenerate, and spinal motor neurons degenerate, and then there's corticospinal degeneration, and then upper motor neuron degeneration. It's like upper motor neurons just sit there and then wait to be degenerated, right? And they don't contribute and nothing. Like they're gonna, they don't care. It's just, it starts from the periphery and gradually goes to the cell body, and then upper motor neurons are gonna die anyways. So we found that that is not the case. Upper motor neuron degeneration is an independent event than spinal motor neuron degeneration. So to build effective treatment strategies, we have to target upper motor neurons and spinal motor neurons together in ALS. And for HSP-PLS patients, you need to focus on the upper motor neurons. Okay. Please read this paper. Actually, it took us maybe 2 years to publish it, and we worked on it for like 6 years. All the experiments were, you give birth to a child 9 months, you publish a paper in 9 years. So it's hard to publish a paper. And Dr. Clive Svensson actually wrote a very nice review about this. Anyway, I'm not going to go into detail, but this is just to show that the upper motor neuron degeneration is actually an indication of the I'm not going to go too much detail, but just to show that we can modulate gene expression in the upper motor neurons. But now we need to be translational, right? We have to develop translational efforts. So that's why I'm very thankful for the Spastic Paraplegia Foundation grant that we got together with Dr. Nicholas Hatsopoulos, and he's working with macaque monkeys. And they're very similar to human in terms of the motor neuron circuitry. And we're trying to understand whether we can develop selective transduction, which means when you give the virus, not all neurons in the brain will get transduced. It's mainly the upper motor neurons, because you know that there's selective degeneration. So you want your treatments to be selective as well, because if you transduce all cells, all neurons, you fix something, but you break at least 10 more. You don't want that, because once the virus is in you, the virus is in you, right? You don't want to, cure one thing and then create 10 problems down the line. So you want to fix it at the cells that need that. In any case, so this is the macaque monkey. This is layer 5. These are the bed cells. These are the upper motor neurons. And yes, I think we can actually transduce the upper motor neurons. Of course, we need more funding. We need more support to see if, this holds true, and then we can write more translational efforts. Okay, so let's go back to our table. Lack of knowledge, check, check, check. Lack of translation, check, check, check. Lack of technology, check, check, check. So we define the problem, and by systematic approach, we check, check, check all of them so that we move forward and by solving the problems. So the path forward, right? What is the path forward? I think there's sunshine at the end of the road. So I think that there will be effective treatment strategies for upper motor neurons. And that's why I'm really working very hard to develop an upper motor neuron center where we will be able to help upper motor neuron disease patients and take them, everyone individually, and understand what's their problem, develop solutions for their problem, and build effective treatments for them, right? And so I need help and support to build the upper motor neuron center. And that will be one of the first upper motor neuron centers in the United States, potentially in the world. And our goal is to develop effective treatment strategies because we laid the foundation. We have the reporter line, we have the drug discovery, we have biomarker studies, we have gene therapy, we have the knowledge from diseased upper motor neurons. So we are beginning to understand why they degenerate. We are beginning to develop mechanism-based drug discovery. We are trying to understand the biomarkers for each patient in terms of metabolites, lipidomics, proteomics, in the serum, in the plasma. So we are working really hard. So now, that's why for the drug discovery, we use the neurons that degenerate in disease as a survival readout for success. Not other disease, but we learn directly from the disease neurons. And that's very unique and that's very powerful. Then we can will have better clinical trial designs. We can include patients based on the perturbed mechanism. And then with effective biomarkers, we can tell if the study is working or not, right? So that clinical trial design will be much, much better. Then we can also have personalized medicine, understand each patient individually and their requirements so that we will have no patient left behind. We can't have a box and try to put all the patients in the same box. No, we need to understand every patient, okay? So then if the patients are so rare that they don't really fit any other picture that we have seen before, then maybe we develop gene therapy approaches for them, just for the gene that they have, just for the mutation that they have, right? So it's not really the gene therapy, you don't really need to, let's say, express the unmutated gene. You may have shRNA, you may have ASO. There are so many other ways now. The technology is also developing. So then at the end of the day, we focus on the neurons. We focus on the disease neurons and we understand why they're unhappy. We speak to them, we ask them, they reply, they tell us why they're unhappy. Then we make them happy. Then when they are happy, the neuron will be functional again. And then when the neuron is functional, then you can actually develop treatment options based on each patient for their unique needs. Then that's why I think that building effective treatment to upper motor neuron diseases, yes, it is possible. And we are moving in that direction. And your help and support, especially to the Spastic Paraplegia Foundation, is much appreciated because they are the ones who support that research in many different areas in the world, right? Not in the United States, but everywhere, in London, in Australia, like whoever is doing upper motor neuron research, they give them grants. But now it is time to build teams so that rather than feeding this fish on this ocean, this fish on this ocean, we actually have a pool and bring all the fish that is going to solve the problem in the same pool and feed the pool. Because when you feed the ocean, it is diluted, but you feed the well-characterized pool, then your the impact for making effective treatments is improved. And that's what we would like to achieve by the Upper Motor Neuron Center. And I will stop here and take any questions that you may have. Thank you so much.
Gosh, Dr. Ozdinler, I don't even know where to begin, but that was an amazing presentation. And as Jackie had said over in the chats, thank you for giving us hope. And, I see so much that, I don't understand all of it, but I know that you do, and I see your passion. And I'm just so thankful that you're there working on our behalf.
Thank you so much.
I'm sure there's some more questions, and if you'll look over in the chat, Fernando has also provided another comment and maybe a question there as well.
Yeah. So
anybody else have a question, get in line.
All right. So I want to say that I like the word hope, okay? But there's one word which I like better than hope, and it is planning. So when we plan better, right, we generate hope. hope by itself is like a balloon, right? It's good to have. It makes you happy. But planning is actually more valuable than hope. So I am trying to make plans so that we all can have hope, right? So I can't be just hopeful and cheerful, but no, I am strategically making plans. So I have like 3-year plan, 5-year plan, you know, and I think planning is really more important and valuable than hope, because hope by itself may not be enough for treatments. I think we need like better better planning, and hopefully we will be able to achieve this.
I certainly agree with you. Does
Yes.
anybody have any other questions that you would like to ask at this time? Go ahead and hit your reaction button and get in line. If you don't mind, Dr. Ozdinler, we have someone over here that Mr. Fernando has put a comment in the section. You might want to take a look at that.
We know that there are some ALS patients that got this condition by exposure to toxic agents or infection agents. Furthermore, these patients don't have typical gene mutations. Yes, very good. Could we explain this toxic spread of agents in their body by a prion-like mechanism? Okay, so, there are many questions here. Yes, so as you know, especially in ALS, some of it is familial and most of it is sporadic, right? So either we don't know the mutation, it may still be unknown, or it may really be not a mutation. So you remember we were talking about proteins and protein bindings and so forth. So for the proteins to bind or to interact, they must have a proper 3-dimensional structure, right? So that one protein is like this, the other protein is like that, or that, like, proteins have different structures. And the protein folding is also very important. And let's say one molecule binds to them. It's a toxic protein, let's say, or a toxin. And then normally the protein must bind like this. But now that you have the protein, it can't do this, it does this. So it's different. It's not L, it's R. It's different, right? So now that it's like this, it can bind. Like, it cannot bind to the location that it is supposed to bind. But if at genetic level, it says, well, you don't have any mutation, like you should be doing this. Well, it says there's a chemical stuck here, and I can't really fold properly. Like, this is how I am now. Like, I'm like this now, and I can't really speak now, or I cannot really talk now, or I cannot move my leg down. So you are stuck at one position that you should not be in that position, and you cannot do your function with that position. But then the genes don't understand. They say, you don't have a mutation, go do it. And it says, I can't. So then it's not that you cannot, you also tell your neighbor next to you, like, I am like this, and can you help me? And then the moment you get close to them, whoop, they turn like you as well. So it's like the prion, like proteins that are in a different format, they make other proteins in the other format as well. So then that can accumulate as well. So it is a possibility. That's a theory. It can happen. And that also, this toxins also explain why this misfolded protein happens in the presence of some toxins. And so I think there's truth to that. And it may also, promote a prion-like mechanism. But of course, which protein it is, what folding it is, what folding structure it is, those are mechanisms that we don't know for each patient. Okay, so what do you think will come first, gene therapy or drug therapy? I think they're both are going to come at similar times, because drug therapy has its own challenges, right? Because inclusion of HSP-PLS patients, they are very rare, and what is the mechanism and so forth. And gene therapy has its own challenges as well, for lack of toxicity, phase 1, and which gene is it also upregulation, downregulation? Because in gene therapy, let's say you have a mutation in the gene. Now you're going to give the wild type gene, right? But sometimes giving the wild type gene may not be the solution, because now you have production of the wild type gene, but you also have the mutated gene, right? So if the mutated gene has a prion-like kink, Now you are giving more fuel to the fire. You're giving them more of that protein, and it's going to add to the kink, and you may have, rather than improvement, you can actually have worsening of the effect. So sometimes, are you going to give the wild type gene? Are you going to have a gene that binds to the mutated form and does, let's say, homologous or homologous recombination? Or are you going to give something to silence the mutation? Or are you going to do a knockdown? So you have to do those studies to see which is the best way to treat at a genetic level, right? And then also, is it really cell type specific? Is it really affecting other cells? And is it okay to affect other cells? So there are numerous questions for the gene therapy as well. What is the site of injection? What is the dose? And how are you going to repeat it? And what is the tolerability? So I think these are two important questions. And I think, we're going to develop them side by side. It's not like one or the other. And it's not like first this, second this. But who's going to touch the finish line first? I think it will depend on the patient population. So for some patients, it may be gene therapy first, and some patients, it may be drug discovery first. All right, what do you think will come? Okay, so I answered that question. All right, good.
But it's pretty remarkable, Dr. Ozdinler, that you guys, that you have drilled down into the very depths of these things and you understand them. Yeah. I mean, to be able to be at this point in our discovery of science and how this all fits together is remarkable, that you guys, are at this point. And of course, we're all getting older and we're all trying to hold on to the planning and the hope. And so thank you so much for all of the hard work and the discoveries that, you have at this point, and I can see that there's so many things that are just right on the edge that we are just about ready to explode this and find some real, therapies or treatments or cures that can make a difference in all of our lives. So it's just phenomenal that you understand, where things are. And you said something very early on in your speech, and it was that, there's no special call from NIH. That really needs to be addressed. And maybe that's one of the things that, we as a foundation, when you talk about planning, we have this last couple of years really drilled down and exploded in growth, with our organization and with our population. And we're finding more people, all the time, to come in and join their hands with us. And really, put forth who we are and what we're doing, and why can we not have someone from, the NIH say, oh, wait a minute, we need to study that. Our population is here and we're not going anywhere. And if anything, we're finding more and more people like us. So, we need to be doing some more planning in those areas as well. And I think, we're at the point where these things that we're now finally hearing about, and we need to find ways that we can elevate that.
Definitely. And, forming teams are very important. First of all, it was like individual people working together. And it was very hard for me because, I was a young female faculty saying upper motor neurons are important, upper motor neurons are important. Oh my God, in a sea of people who said, who cares about upper motor neurons? Who cares about upper motor? And there was actually a time in my career, I did not publish a single paper for 6 years. And for a junior faculty, if you don't publish anything for 6 years, you're dead. Like you cannot get grants. You cannot, you were basically dead. So then they told me, stop working on this, just do something else. But in that 6 years, I actually looked at more than 600 genes that are expressed in the upper neurons, trying to make the transgene. And I said, no, we need to go down. Because, there are, let's say you lost your car key in a spot, but then there is light post far away. You wouldn't look for your keys under the light post because it's bright. You would look for your keys where you dropped them. But then you cannot say, oh, but there's no light there. Yes, but if you look under the light post, you're not going to find it. It may be easy to look under the light post, but you're not going to find it. So my theory was that let me bring the light post where I dropped my keys, okay? It took me a very long time to bring the light post where I dropped the keys, because we had to make the reporter line, we had to make them fluorescent, we had to find the keys, but once you find the keys, then you open the door. And all the people who started working with cell lines, this stuff, because it was easy to grow, because it was less expensive, because it was convenience, because it was this, but then results may not be as translational. But now that we actually open the key, we're going to drive the car. So that's our hope.
That is our hope. And we thank you so very much. And for taking time this evening to help us to understand this. And we would certainly, I'm sure, get a thumbs up from everybody on the screen that we would love to have you back.
Thank you so much. And thank
Absolutely.
you for your support. And, you know, I also want to say the Spastic Paraplegia Foundation is very unique, that they support the upper motor neuron research, and all the patients, they must do everything they can to support the foundation, because they not only support the research, but they also support the patients. And I think they're doing a phenomenal job, like, you know, with the symposium, with the support groups, with giving funds to the researchers, with helping patients with their needs. And, you know, if we are, you know, together as a family, as a community, then nothing can stop us. But if we don't get together, like the neurons in the circuitry, right? If we don't form that circuitry, it's going to fall apart. So we all have to pay our dues so that we are a family, and then we are together in this fight. Because one neuron by itself can do nothing, and one person by itself can do nothing. But when we form the circuitry, and I think Spastic Paraplegia Foundation is doing that for the upper motor neuron biology, for the upper motor neuron disease, for the patients, for everything, then there is hope, then there's plan, then there's future, right? So I'm happy to be talking to you today, and I hope that we will form those teams, and I hope that we will celebrate together. And I really want to see HSP and PLS patients say, well, I had HSP when I was young, you know. Let's let that happen.
You know, I could not have paid you anything to have given that perfect commercial for us.
Yeah.
So, and of course, it's like,
It's
you know,
not really a commercial, it's truth, you know that.
I know, yeah, it is, and it's like you're, you know, you're really kind of preaching to the choir, right? I mean, we already all, I kind of know this, but it's so refreshing to actually hear you say that for us, and I greatly do appreciate that. And I think we have got a wonderful family, all of us together, and even many that can't be with us. And I know we have had some new friends that we've made this week that we're bringing into the family. And, you know, this being our awareness week, and I know Fernando had made a comment here about that he didn't hear your last answer, but please don't fret, because we are recording this, and once our tech people, Hank, Dina, that once they get all of this information put into the processes, we will have all of this uploaded to our YouTube channel. And I would certainly love to invite all of you to go and watch the YouTube channel, where you will be able to see Dr. Ozdinler at our Nashville conference, and you'll be able to hear her presentation that came from that as well. So please take time to click that follow button on the YouTube channel and make sure that you guys, you know, stay in tune with some of the things that we're putting on there. And please let me remind you that we have all of the social media platforms that are out there, and we would love for you to be a part of those. And there's all kinds of dialogue that's just constantly going on. People are helping, people are talking, new folks are getting into, you know, understanding that they've been diagnosed, and, you know, they all need support, they all need help. And one of the things that I would like to invite you to know is that, you know, this is our awareness week, as I said earlier, and every night this week at 6 p.m., just like tonight, we have, you know, the activities that we're doing, and we were hopeful that some of the other doctors were going to be able to commit to coming in and being a part, you know, each night as well. But there's been so much conflict that we hadn't been able to get some of those others scheduled. But I do have a gentleman tomorrow at 1 p.m. Central that's going to be talking with us, Chris Pace from Utah. He is going to be talking about self-care and how that really interacts with having a chronic illness or a disability, and some of the things that we should be thinking about and things that we should consider when we're dealing with a major stress. So I would love to invite all of you to come back tomorrow at 1 o'clock Central, and we're going to pop this Zoom program, and we're very thankful for the Zoom program, but he's going to be sharing some information with us. And then, as you guys note, if you will, on our social media platforms, he has provided to me a book that he is going to be able to have available, a PDF that you can actually download. If you will, find that information on our website and on our social media platforms, and you will be able to have that information so that you can go through that workbook with him.
Wonderful.
Great.
Yeah, so I'm not sure what happened to the screen. It seems like something kind of zipped in, but you guys can still hear me.
Yeah.
I'm not sure if you can still see me,
Yep.
but please make sure that you guys check out the social media platforms, and we will have links to that book. And as I said, there is a PDF that you can actually download and see that information, and we'll be able to share that information with you. But it's the workbook, real quickly, it's called The Chronic Illness Workbook by Patricia Fennell, Fennell, and you'll be able to grab that information off of, like, you know, the Google or one of those platforms, and you'll be able to download that as a PDF. But anyway, I appreciate everyone joining us tonight. I'm going to go ahead and close us off, and we will be able to have an opportunity to meet again later in the week. Dr. Ozdinler,
Thank you.
blessings to you. Stay
Thank
healthy,
you.
stay active, stay planning, and stay hopeful for us. So I certainly appreciate that.
Thank
very
you,
much.
Dr.
Thank
Ozdinler.
you.